In this next section, we're going to be recapping on some of the functions that we looked at in the first workshop around data assembly and thinking a little bit more about how those functions can be tuned or adjusted to suit different kinds of data. Specifically, we're going to be recapping on the calc flowstats function, which we use for summarising the hydrological data, and also the join HE function, which we use to join together our biological and hydrological data. Starting off with the calc flowstats function. You may remember from workshop 1 that we can use the HE toolkit to import a time series of daily mean flows for one or more gauging stations, but that we typically want to summarise that data to be able to characterise how key aspects of the flow regime have changed over time at each site or station. The calc flowstats function provides a very flexible way of doing this. It allows us to define a moving window which breaks up our time series of data into a sequence of time windows. It then calculates a broad range of different summary statistics for each of those time windows at each site. We've got a lot of flexibility or control as the user about how we define that moving window. We can control the start of the moving window, that's the first day of the first window, the width of the window, and also how much the window steps on each time. And as shown on the slide, we can use it to generate contiguous time windows, for example, annual time windows, non-contiguous time windows. For example, if we wanted just to focus on summer flow conditions, we could have a time window that defines the 6-month summer period in each calendar year. Or we could have overlapping time windows. This is where the time step is small relative to the width of the window. And this gives us a very relatively granular way of describing the flow conditions over a given time period prior to a particular sampling occasion. Now, when we're using this function, we have to think about those different window settings. So we have a lot of control over the definition of that time window. We might, for example, choose a relatively wide window if we think that our biological receptor of interest responds quite slowly to antecedent flows and or we want to consider the long-term chronic effects of changes in flow. In that situation, we might choose a window which spans several years and provides sort of an integrated measure of the flow conditions over an extended period prior to each biology sample or survey. Alternatively, if we're interested in short-term acute effects of flow, so relatively short-term drought or flood events, for example, then we might choose a relatively narrow window which is able to home in on the flow conditions that have been experienced relatively recently prior to sample collection. Another consideration is to think about whether we're interested in just the recent flows and or the lagged effects of earlier flows in previous or older months and years. In that situation, we might choose, for example, a set of contiguous or overlapping windows, but then take the flow statistics from windows relatively recently in the past, but also further back in the past. And that can provide a way of understanding whether the ecology is sensitive to relatively recent flow conditions or whether there's a sort of cumulative long-term effect of flow conditions that have happened further in the past. For some types of analysis, for example, when we're working with data on fish, particularly salmonids, for example, those species may have very defined life cycles, and there may be particular points in the life cycle when they may be potentially very sensitive to flow. And I'm thinking, for example, spawning periods or periods post-emergence when you've got very small fry, which can be very sensitive to flow conditions or very sensitive to flood disturbances, for example. In this situation, we could choose to define windows which allow us to summarise the flow conditions during those very specific points in the year when we expect sensitivity to be greatest. So again, by setting the wind start date and the wind width, We can define, for example, a period during an autumn spawning time, or during a post-emergence time later on in the spring, and allow us to capture and describe the flow conditions during those very critical periods. Another consideration is to think about what we're going to use the model for. As well as thinking about sort of the ecological aspects, we also might want to think about it from a policy perspective. If, for example, we were using hydro-ecological models to define a local flow target, then it might make sense, for example, to do that over an annual time period so that the results might be comparable with the default flow targets suggested by the Environment Agency's Environmental Flow Indicator. There is no right and wrong way of doing this. It's very much horses for courses. So when using the calc flow stats function, it is important to think about the ecological considerations. So what is your biological receptor? How might it respond to flow? And also think about how you might then interpret the results which are based on that data. And the flow statistics or the time windows you choose will be a mix of those different considerations. As well as thinking about the time window and how wide or short and which parts of the year it covers, we then have another choice to make in terms of which flow statistics we choose to use from the calc flow stats function. You may remember that the function provides us with a very comprehensive and diverse set of 60 flow statistics for each of those time windows. Those range from very simple statistics such as the mean, or minimum and maximum flow, different percentile flows, and also statistics which describe the frequency and duration of high and low flow events, and also the timing of events through the course of the year. There are far more flow statistics here than you would probably ever need. And it's really important to be aware that a lot of these statistics will be quite strongly correlated with each other, as you might expect. So if higher flows are high during a particular time window, then it's quite possible or likely that low flows will also be higher than normal. So there's likely to be a correlation between these different statistics. There's also a certain degree of redundancy in that some of these statistics may essentially be just slightly different ways of summarising very similar aspects of the flow regime. So again, it's worth taking your time to familiarise yourself with the range of statistics produced by the function, and then be choosy about which ones you take forward into your analysis. Remember also that flow statistics which describe the number of events, so for example the number of drought or flood events during a particular time window, and also statistics which describe the timing of for example, high or low flow events, those will be directly comparable across sites because they are either simple counts of events or the timing statistics are based upon the day of the calendar year. Conversely, statistics which are based upon absolute magnitudes of flows and so therefore measured in units of cumex, those will not be directly comparable across watercourses of different sizes. And in that situation, the calc flow stats function provides standardized z-score statistics for those or versions of those statistics, which are dimensionless and therefore comparable across sites. The details of how that's done are covered in workshop 1. When choosing which of the flow statistics to use, we need to think about which aspects of the flow regime are most likely to be important ecologically. So here we may have to draw on previous work, research, or our own expert judgment to think about whether it's maybe the low flows which are likely to be most critical or influential from an ecological point of view. Maybe it's flushing flows are important for removing fine sediment. Maybe we're dealing with an intermittent system, in which case it's the frequency and timing of drying events which might be most important. Or if we're dealing with species which have a very migratory or mobile and there's barriers in our catchment, then defining flow statistics which can help to define the passability of those barriers may be most critical. Again, there's no right and wrong way of doing it. It's horses for courses, and you can choose different flow statistics based upon your own understanding of the ecological system. Just as when we were thinking about defining a moving window, though, you can also think about this from a sort of a management or policy perspective. Are we, for example, interested in percentile statistics, which may have some relevance for interpreting whether or not FE flow standards are appropriate? If we're dealing with compensation or hands-off flows, then we may want to, for example, use statistics which describe the number of times or the number of days that flows fall below a particular set compensation flow or hands-off flow. Again, maybe we're thinking our study system is rivers downstream of an impounding reservoir, and we may be interested in the flow variability, in which case statistics which describe the number of spill events from the reservoir could be a very useful and ecologically relevant statistic to use. If you're a bit bamboozled by the choice of statistics or not quite sure which ones to use, then a good starting point is to try and use a mix of statistics which describe both high and low flow events within the hydrological time series. So that's hopefully gives you a sort of a few ideas or some advice really in terms of how to use the calc flow stats function. The next step in the process is to join our biological sample data with those flow statistics. And to do that, we use the join AG function. Again, we talked about the use of this function in the first workshop, but just to recap, the join AG function allows us really to join the data in such a way that we can describe the antecedent flow conditions prior to each biology sample. To do that, the sequence of flow statistics for the sequence of moving windows are numbered. So those time periods or windows are labelled relative to the date of each biology sample. So in the examples you can see on screen at the top, we've got a series of contiguous flow statistics for each calendar month. We can then define the lag 0 time period as the one for either the most recently finished time period, which we call method A, or the most recently started time period, method B. So in the example on screen here, we can see that we have a biological sample collected in the middle of September. And using rule A, the most recently finished flow time period is the August time period. So that's lag 0. And then we count back from that. So July is lag 1, June is lag 2, and so on. Using rule B, the most recently started flow period is September. So that becomes lag 0. And again, we count back. August is lag 1, July is lag 2, and June is lag 3. In this way, we have a nice, simple system for defining which time periods we want to pull in flow statistics for to then correlate or regress with our biological sample data. And this gives us control over whether we want to look at relatively recent flow history or flow conditions experienced further back in the past. The second example at the bottom of the slide works in exactly the same way, except this time we've defined overlapping time windows, but the principles are exactly the same. So the question is, when we're using this function, which of these rules, rule A and rule B, should we use? They have pros and cons. Again, there's no right and wrong choice. Rule A, which happens to be the default option within this function, means that we ignore all flows after the biology sample. So using rule A, if our biological sample was collected on the 15th of September, for example, then our flow statistics for lag 0 are for the August period. This means we actually overlook some very recent flows in the first 2 weeks of September. It does mean that our flow statistics don't include any flow data or flow conditions that occurred after the collection of that biological sample. Conversely, rule B ensures that our flow statistics cover all the flows right up to the date of the biology sample, but it can also then potentially include some flow measurements which occurred after that sampling date. So again, if we assume that our biology sample was collected on the 15th of September, then taking flow statistics for that lag 0 month means that we cover the flow conditions in the first 2 weeks of September. We also cover the flow conditions in the 2 weeks after the biological sample, which obviously couldn't have influenced the ecology at that particular sampling time. When we're using the join HE function, we can have any combination of lags. So that gives us the flexibility to investigate either very recent flow conditions that have happened further back in the past, or a combination of recent and older flow conditions. So we can look at the total or cumulative effect of flow conditions at different points in history. We can also choose whether to define flow conditions for a fixed time period. So, for example, we might want to characterize the flows in a particular month or in a particular, say, summer time period, in which case we might use the function to join our biological sample data with the summer flow conditions that have occurred in the previous year and the year before that. Alternatively, we might be interested in the sort of relative in the flow conditions over a relative time period. So for each sample, for biological sample, we might use the function to join in the flow statistics for a 6-month period prior to that sample being collected. To do that, the overlapping flow time periods are particularly useful. Here we're taking each biological sample and marrying up with the flow statistics for say, the immediate 1 or 2 or 3 month period prior to that. Again, that gives us a nice flexible way of describing the influence of flow conditions over a particular relative time period. Join type argument also gives us 2 different ways of joining the data. This is described by the join type argument of a function. The first option is to set join type equals add flows. Here we take our table of biology data, as shown at the top here, and a table of flow statistics, and we join the flow statistics to the biology data. That gives us an output which is shown in the table with the blue border here. The number of records in the output table is the same as the number of records in the biology input table. So here we have 2 different biology sampling records, and for each of those we join in the Q95 flow statistics for the lag 0 time period. This gives us a data set which can be used for calibrating a hydroecological model. We have a column containing our response variable, our biological metric scores, and we also have columns containing 1 or more flow statistics for the antecedent time periods. Notice that there's no missing records in here, so we have a data set which is ready to go straight into a hydroecological model. But there are situations where we might want to flip it around and instead set join type equals add biology. Here we take the table of flow statistics as our starting point and then add in the biological metric scores where those are present. The output table is shown here with the blue border. Now we have the number of records in our output table matches the number of records in our table of flow summary statistics. Where we've got biological data, it gets joined in, but where there's no biological sampling, we get an NA. Joining the data in this way allows us to create a data set which is useful for visualization. We looked in the first workshop at how we can use this kind of joined data set to produce a hydroecological validation plot, a HEV plot. And we'll also see a little bit later on in this workshop how we can use this data set to explore the coverage of the data using the plot range of flows function. So again, that's just a little bit of a recap really on some of the functions that we looked at in workshop 1, but just elaborate slightly just to emphasize the control and the flexibility we have with those functions and the need to tailor the way in which we calculate the flow statistics and join them to our biology data to suit the needs of each individual exercise or project. If you would like to have a go at putting some of those functions into practice, then follow the link to the POSIT Cloud project that accompanies this workshop, and there you will find some case study data sets and some example R scripts which provide a template for the code that you can use to calculate flow statistics and join them to your biology data. So if you'd like to have a go at practicing that, and indeed maybe playing around with some of those settings to the functions to see how they influence the results, then please head over to Practical Exercise 1 now.